My name is Brandon Simmons. I'm one of the general partners here at Prime Movers Lab, and I'm really excited to welcome what looks like a pretty big and gathering audience to the second longevity webinar that I've had the good fortune to help lead. Many of you may remember last year in what for me was our first longevity webinar, we featured a couple of real legends in this space, Aubrey de Grey and David Sinclair, along with Mark Allen, the founder of Elevian, one of our portfolio companies that is focused on longevity. So that was more of a high-level discussion. And today we're going to go deeper and really zoom in on one of the most exciting areas in longevity investing. So before I turn it over to my partner, Jyothi, who is going to introduce herself and then also get the real stars of our time today going, I want to briefly welcome, I see a number of our founders in the audience, and I see many of our limited partners joining us. And we are so grateful and fortunate to be in partnership with all of you, and love it when you join us. For anyone who is joining us for the first time, at Prime Movers Lab, we invest in breakthrough scientific inventions that we believe can transform billions of lives. And I think I said this in our last longevity webinar, but I really feel like we are on mission when we're working in longevity. It's an opportunity for us to very directly transform billions of lives. So with that said, I'm going to turn it over to Jyothi to introduce herself and give us some rules of the road for this webinar and get our panelists going. Thank you.
Thank you, Brandon. Hi, I'm Jyothi Devakumar. I work with Prime Movers as the principal biologist and partner. I have a PhD in biotechnology. I have experience in the big pharma type drug discovery. I've managed several C-level positions in companies in the verticals of regenerative medicine, cell therapy, and longevity. I have experience in several consulting assignments, academia, and also in angel investing. Now, as a part of PML, since April, I'm focusing on our investments in longevity and regenerative medicine. The focus of this webinar is to understand one of the most fast developing areas of longevity industry, which is senescence. A cell is a massively complex entity in which a huge network of processes enable it to perform its function and also maintain its own stability. During life, this highly coordinated homeostasis sometimes gets disrupted, leading to cell death or cell becoming cancerous or to a cell becoming senescent. In brief, a senescent cell is like a zombie cell, which is actively toxic to its environment. Now, when it was discovered that the senescent cell burden increases with age and contributes actively to several side effects of aging and chronic diseases that we experience with age, the next step was to see if removal of these cells would be rejuvenating. The initial success was so dramatic, it captured the attention of scientists, founders, and investors, creating a fast-growing, highly investable sector with the potential to address several chronic diseases of aging. Today, our aim is to get some of the best minds in the field to help us understand the super exciting area, both from a scientific, industry, and pharma perspective. Now, going into some tips on housekeeping, we will keep this to 1 hour. We will use the chat and Q&A feature to ask questions. The questions could be general to all the panelists or specifically addressed to one of the panelists. We will leave 15 minutes at the end for questions. And don't forget to join us next week. Same place for Liz and Christy, our partners discussing high-speed flight. And I'm grateful to be a part of this stellar panel. I request each of the panelists to give a brief introduction. What we would like to understand from you all is, how are you contributing to the field of senescence? First of all, who you are, how are you contributing to the field of senescence? And what brought you to this field of senescence specifically and longevity at large? And what inspires you to continue the fantastic work you're doing in the field? Paul, let's begin with you.
Yeah, thanks, Brandon. Thanks, Jyothi. My name is Paul Robbins. I'm the Associate Director of the new Institute on the Biology of Aging and Metabolism at the University of Minnesota. And my lab, at least for the last 10 years, has been working in 3 different areas of developing approaches to treat or at least extend health span. And one of those is trying to understand what types of cells actually drive aging. So we're using mouse models where we can induce senescence or DNA damage or other types of damage in different cell types and tissues and see how they contribute to lifespan in mice. We've been developing drugs, senolytics that actually will kill different types of senescent cells. And I can tell you that there's no one type of senescent cell, so not one drug is going to treat every type of aging pathology. And then lastly, we've been collaborating with Albert Einstein using their centenarian cohort to use the genetics of centenarians to identify rare variants that are linked to longevity. What's interesting is that these variants appear to be linked to senescence, that they seem to reduce the level of senescence. So we really have 3 different approaches. We're trying to tackle the role of senescence in driving aging. And how I got into this area was I turned 50, so that was the first thing. And then the second thing is I bought into the geroscience hypothesis that I work in diabetes or have worked in diabetes, cancer, arthritis, muscular dystrophy, and the same pathways seem to be going up or being altered in all of these diseases. And then started to think, well, the biggest risk factor for these diseases, at least not the genetic diseases, but ones you acquire with age, is actually aging itself. And so I really brought into the concept, the way to treat these diseases was to treat aging itself. And so I am completely committed to this area because I think this is the way we make the biggest difference in human health over the next century or so.
Marco?
Thank you, Jyothi and Brandon, for the invitation organizing this event. This is amazing. So I'm Marco Quarta. I'm the co-founder and CEO of Rubedo Life Sciences. I'm a serial entrepreneur. I started other companies in this longevity therapeutic space. And I'm also a scientist by training. I have a PhD in aging neuroscience. I did my postdoc at Stanford with Tom Rando, which is another early pioneer. My mentor is the one who pretty much brought this idea of the parabiosis, right? Putting together an old mouse and a young mouse and sharing blood. There's something in the young blood that can make old mice somehow rejuvenating, even better, healthier. It's also true the opposite. The young mouse lives less and is worsening their condition. Anyway, that brought me in my training in stem cell biology, aging, regenerative medicine, and I've been at Stanford for more than a decade, driving my group focused in aging and regenerative medicine. And where I'm doing, I mean, I now fully committed in Rubedo. I left academia to really translating my science into something that I can give back to people. So that my goal is really developing therapeutics that can help in making this idea of longevity therapeutics and contributing to the field possible. Now, to Paul's point, what we do at Rubedo is developing as a new generation of pharmacological treatments that are, let's say, smart drugs that are smart or special because they don't work unless you need them, right? So the idea is making drugs that are very selective, that take into account, like Paul mentioned, the heterogeneity, and we have different verticals. So the most advanced program are senolytics. So drugs that can remove the burden of senescent cells, but which type of senescent cells and where and how, the pathological one, there are also the good ones. Senescence also plays a positive role. That's very important and critical for have a successful therapeutic in the future. The other important aspect is making drugs that are safe, tolerated, especially in older people that don't, side effects and toxicities are not tolerated in older people with comorbidities, frailty, many conditions that maybe younger people do. So we really thought a lot about making those compounds. Some are prodrugs, so drugs that are inactive and are activated only in your target cells, in the cells that you need to remove, the bad cells, and you're protecting the good cells. So that's what we do and how we contribute into the different modalities that are around in taking advantage of senescent cells as a therapeutic opportunity. Now, why I'm doing this? Well, my interest in longevity actually starts very, very early, actually a very young kid. I was actually 5 years old when I told to my mom that I want to cure aging. That's something that my mom actually reminded me more recently. But that is true. And I started my first lab at the age of 7. Luckily, many scientists in the family, meaning that I started to borrow or take, chemistry gears, microscope, and I really started my personal journey since then, and I'm still doing it. I'm less naive now, but I think that we can target aging to treat age-related diseases. Yes, that's a reality that we will probably see soon.
Yeah, thank you, Marco. Now we move to the most impactful person on the panel in terms of creating exits, and the person with the most beautiful name, Angele.
Thanks, Jyothi, and thanks, Brandon, as well. So again, I'm Angele Mackey. I work in the corporate business development group at Eli Lilly, but I'm based in the Bay Area. I've been doing business development for 17 years now. Trained as a scientist, though. I did a PhD in biological chemistry at Stanford, but never practiced as one in industry. I went right into business development. And I really love this job because the way I describe it to lay people is that I get paid to meet people and learn new science every day. So it's really great. But the reality of the job is that we are out there looking for new technologies and sourcing innovation for the pharmaceutical company that we work for. That's what the job entails. And so in my particular role, the particular group I'm in is that we focus on early stage technologies and early stage companies. And one of the primary goals of my particular group is to stay abreast of cutting edge technologies. So everyone in my group has a PhD. And so again, we're charged with staying abreast of cutting edge technologies and sorting out how these may translate into investments we may make or partnering opportunities, M&A opportunities, all sorts of things. So that's what I do. What brought me to the field of senescence is that obviously it's a burgeoning area. A lot of companies are being formed in this space. And it's one that really spans a lot of different therapeutic areas. So it could be a very high interest to pharma. And I think it's still very early days in the science, as we'll get into in a bit. But I'm continuing to monitor it because it's also a very fascinating area. It affects everyone. It's not like some obscure metabolic disease or genetic-based disease that you don't know anyone that has it. This affects everyone, so it's very personal. And who doesn't want to stay younger or not age as quickly? So I guess there's a personal element for me, but also it's just fascinating new area of science as well. Thank you.
That was great. Thank you all for those wonderful introductions. I'm going to ask the first question, and I'm going to ask you, Paul. Well, first, I love how you said that you got interested when you turned 50, and then Marco just massively up-leveled on you, saying he got interested at 5 years old.
He's a lot smarter than I am, obviously. A lot more motivated.
Come back to that. I'm always a bit overwhelmed when people I know in this Prime Movers Lab world announce that they've been doing something since they were, the age that I was learning how to throw a ball, probably. But let's move on. Paul, so Jyothi gave a little bit of an introduction to senescence at the beginning, and I think it will be valuable for our audience to hear from you, kind of further laying some foundational background. So maybe if you would just spend a few minutes with a kind of simple explanation to frame us, what is senescence and why is it so important?
Yeah, which actually is not that easy to do. We wrote a review with many experts in the field for Cell that was published a year and a half ago, and it was clear with all the experts providing advice on how to define a senescent cell and how to characterize them that there's no consensus. But in general, a senescent cell is a cell fate, or at least senescence is a cell fate. So it can be driven by DNA damage, which can be caused by telomere shortening, oxidative stress, genotoxic stress, epigenetic dysregulation, or even oncogene activation that stimulates uncontrolled growth, lead cells to enter this senescent state where they leave the cell cycle potentially irreversibly. So they can't proliferate, they can't contribute to cancer. And what makes them both good and bad is they start to secrete factors to attract the immune system to come clear them, at least the bad cells. So they start secreting a number of inflammatory cytokines, metalloproteinases that can break down the tissue to allow immune cells to infiltrate in. They release reactive metabolites. They release extracellular vesicles, a whole spectrum of things that they secrete. That when you're young, the immune system can help clear them and keep the senescent cell burden low. But as we age and our immune system starts to fail, these cells can accumulate. And this can lead to chronic inflammation and even higher inflammation at specific sites. So these cells have been identified at different disease sites, whether it's the joint for osteoarthritis or the lung for pulmonary fibrosis or the brain for Alzheimer's disease. And so the thought is that these bad senescent cells, if you could clear them, that they would improve pathology, both locally and systemically. And that was demonstrated, as Jyothi referred to, in several papers from the Mayo Clinic, Jan van Dersen and Jim Kirkland's groups, that when they genetically modified a mouse so that you could clear senescent cells with a drug, the mice improved. They aged in a healthier way. And that really caught the eye of everybody, both in academia as well as in pharma, that this may be an approach that we can treat numerous diseases and conditions. And I should just point out that you don't necessarily, we're not trying to treat just diseases, but there may be conditions. For example, old tissue that's used in transplants or could be used in transplants, like kidney or liver from an older donor. It appears that clearing the senescent cells from the tissue prior to transplant improves function after engraftment. So many different functions or approaches for how you could use senolytics to treat human disease or improve conditions. Hopefully that was not too complicated.
That was fantastic, Paul. Thank you. Now that based on what you said, it's becoming a reality. Clearance of senescence is a reality. And now, what kind of clinical trials are ongoing in this field, and what kind of progress has been made? Could you please share some of your thoughts on that?
Yeah, it's a great question. So what's amazing about this field is how quickly it's advanced from preclinical studies to clinical trials. And one of the reasons why that's the case is that senescent cells upregulate anti-apoptotic pathways. So although they're damaged, they actually upregulate pathways to keep them from dying. You would expect them maybe they apoptose, but they don't. And the pathways they upregulate are similar pathways that tumor cells appear to have upregulated so that they don't die as readily. And so what it means is that many anti-cancer drugs can be repurposed to clearing senescent cells. I'm not saying these are the best senolytic drugs, but they do work. And so it was an easier path to the clinic for repurposing existing anti-cancer drugs, such as navitoclax, which is yet to make the clinic, but is one that's being developed. A combination of dasatinib and quercetin is the one that is in most of the clinical trials. And then Unity Biotechnology has another BCL2 inhibitor that they moved into the clinic for macular edema. And they had an unsuccessful trial using something that stimulated p53 activity in the joint for osteoarthritis. But I think the number of trials is somewhere around 20 at this point, ranging from actually a trial that I'm involved with to reduce mortality in older COVID patients. We've had a paper in Science recently that showed that the senescent cell burden seems to contribute to mortality. So there are several COVID trials ongoing. Trials for idiopathic pulmonary fibrosis, for Alzheimer's disease, for type 2 diabetes and metabolism. There's a trial starting at St. Jude's for long-term cancer survivors. So these are children that have been treated with chemotherapy or whole body irradiation. They have an increase in senescent cell burden and they are aging faster. So this is a population that's very amenable to this approach. So there are trials starting for cancer survivors. So a number of different indications that there are clinical trials ongoing using kind of repurposed drugs. So what Marco's talking about is not yet entered the clinic, but has a lot of potential. And I think there are going to be much better strategies than what's currently being used to kill senescent cells. The drugs that are in the clinic are ones that appear to be safe. They appear to have some efficacy, but I think they can be improved upon tremendously. And that's what Marco and others, including my lab, are currently working on.
Thank you. So now we turn to you, Marco, to give us an industry perspective, where we are, what kind of technologies are being developed, the difference between maybe xenomorphic, xenoblockers, and senolytics. And this question is open to everyone on the panel. once Marco is given his perspective. Marco?
Yeah, for sure. That's also a very important question. There are different modalities, right? And I think they all have potential. Some might be lower hanging fruits. Others may require better, longer understanding. They might be ready yet. As you mentioned, so one way is senolytics, which means killing senescent cells. So cells that already they became zombie cells. Some people call like that. They're like they stick around, like Paul said. They're toxic. They release those factors that drive chronic inflammation, fibrosis, etc. So, it's known that you need to get rid of them because the body at some point is losing capacity to clear them. When you are younger, we actually do, and senescence is transient and is important, like support regeneration of tissues, like wound healing or tissue remodeling. So it's an important mechanism. It's called pleiotropic because it has multiple double functions. It could be good, but it could also be bad, and becomes bad when it becomes chronic. And then you need to have the body to get rid of them. So senolytics are different classes. And like Paul said, the first generation of senolytics are repurposing drugs that not surprisingly come from the oncology space, because senescence is also a tumor suppressant mechanism. So it's a way that the body has when a cell accumulates too much damage or stress to block the cells to prevent to develop into something worse like a cancer. But sometimes they can escape from this mechanism and move into an oncology program. But what it happens, and when the senescent cells accumulate over time, then they become pro-cancerogenic. There are enough, and that those factors that release actually are promoting cancer spread development. So older individuals have a predisposition to develop maybe more aggressive or frequently secondary tumors. So getting rid of them very specifically is important. And so not surprisingly, I was saying the first generation of compounds have been found in the oncology space because they share metabolism and function, but they come with the cost, which is, as I was saying earlier, side effects and toxicities. The cancer drugs have been developed to kill very quickly highly proliferating cells before they make a tumor or become too aggressive. Senescent cells don't grow, they just stay there. So the good thing, we can take advantage and time of this state to clear them. But the paradox is chemotherapies, radiotherapies, also certain immunotherapies with the adjuvants, because of their toxicity, because they damage the good cells too, they drive a premature aging in these patients. So literally, when we are familiar with that, like chemotherapy leads to losing hair, and people develop all sorts of side effects at any age, like pediatric cancer survivors, so kids, they will develop age-related conditions in their 30s, like cardiomyopathies, like 30 years sooner than the average person. They are chronically inflamed, and cancer survivors have lots of senescent cells that result from their therapies. So it's important to make senolytics that are, as I was mentioning earlier, that's like our effort is making healthy, rejuvenative chemotherapies that can, kill the right cells, but also protect the good ones. So that's one modality. The modulators are drugs that are designed to suppress the release of these bad cells. So the senescent cells are still in your body, but they can't release those cytokines, those metalloproteases, those factors that are pro-inflammatory, pro-fibrotic, pro-cancerogenic. The problem with all that, you have to keep taking drugs because the moment you stop, those senescent cells, they start doing it again. And the problem with that is that senescence can also be positive. So you don't want to suppress senescence constantly. So again, I think the lower hanging fruits is senolytic in that sense because you are very accurate and selective, and so you can remove those and you avoid secondary effects, and you can decide when to remove them, let the body heal and generate senescence when it's useful. Then the xenoblockers are drugs that are instead thought to prevent the generation of senescent cells. It's a different modality. What happens if you can avoid accumulation of senescent cells before that occurs? And I think that's also potential in certain areas. I think is an earlier stage. There is more probably to understand. For example, when is good to prevent it? As we said, senescence is important. It needs to be removed at the right time. Not sometimes you need to have senescence. Otherwise, the result is you get a worsening of your condition. And so that's probably a general area. There are other strategies, modalities that people are developing, such as a sort of a gene therapies, like trying to treat cells so they can kind of induce senescent cells to die, or stimulate the immune system, or using immunotherapies to do that. Again, these are all valuable strategies. I think there is more biology to understand in general. The field is more advanced in the senolytic, like you can fine tune and directly target your right cells, but there is a need to move to a new generation of senolytics that are like safer and more targeted and selective. And that's where we are putting all the effort in how to do that and how to translate this in a pharmacological treatment that can have that level of safety and efficacy and selectivity.
Thank you, Marco. Paul, would you like to add to what Marco alluded to?
No, he did a superb job summarizing kind of the state of the field. Because clearly stimulating the immune system will lead to clearance of senescent cells. People are talking about vaccines against senescent cells, which are probably not the right approach, but Scott Lowe has worked on CAR T cells to see if you could clear senescent cells. So clearly a number of different approaches. I just like to make the one comment I should have made earlier. So aging is thought to be driven by different pillars of aging. And so whether it's mitochondrial dysfunction, loss of stem cell function, loss of proteostasis, inflammation, they're all linked. And so one of the things that's been seen is if you clear senescent cells, you see endogenous stem cell function get better. You see metabolism be normalized. You see things like mTOR, which is a target of rapamycin, it goes down. So all these things that you would like to see improve. And so this is one of the pillars, but there are other pillars, and Marco referred to the fact that it sounds like they're doing things besides senolytics. But what's intriguing is that reducing the senescent cell burden affects all these other pillars. And then we published recently, Marco talked about heterochronic parabiosis. We published that if you do heterochronic parabiosis, that actually there's a reduction in the senescent cell burden in the aged animal. So they're clearly soluble factors, whether it's immune cells or other factors, but they're things which can reduce the senescent cell burden. And then there are things in old animals that can drive senescence in the young animal. So, again, all these things are linked. So I think that by understanding what type of senescent cells drive disease, understanding how to clear them, we're going to see improvement in many different areas. But yet, caloric restriction, exercise, other things actually reduce the senescent cell burden. So there are a lot of ways that we can affect all these different pillars. So very interesting area. I think it's just beginning. We're just scratching the surface, but I think it was Ned Sharpless who commented that because of the speed of the way senolytics have gone from mouse models into the clinic, we will know within 5 years whether they work. And for understanding, a drug working on aging, to understand that in a 5-year period is pretty remarkable. Now, we won't know long-term benefits, but we will know if these drugs actually reduce senescence and improve pathologies or human condition, I think, within the next 5 years, which is pretty remarkable.
That's great. Thank you, Paul. One quick reminder before I ask the next question to the audience. Thank you for questions. We'd love to grow a backlog of questions. I see a great question from Jason that we'll get to soon, and would love to see some more. So I have a question, Angele, for you. We're investors in this space, as are you, and I would love to get from you some guidance from your perspective in pharma on, I guess, on two things. One, what are you seeing right now in specifically this space of senescence, or more broadly longevity, that you think is ripe for investment right now? And then the second part of the question is, what do we need to see happen before we start to see a lot of M&A activity, a lot of acquisitions taking place in this space?
Sure, yeah. I don't actually know if there's anything that, I think it would be tough for a big pharma to make an investment right now, unless they had a champion within the organization that was very bullish on senescence. One of the disconnects between the field of senescence and the smaller companies that are working on it is that big pharma is largely organized around therapeutic areas, and senescence is an area of biology. So where I think the two will meet and connect and be productive is if there are specific therapeutic areas or indications in which senescence has, slowing down senescence or stopping senescence has a big impact, and that's what will get pharma interested. So I mentioned earlier that the intrigue with senescence for us is that it spans multiple therapeutic areas, probably all of them, at least all the ones that my company is working in or playing in. But so I think that's a big challenge, and I think that's why we're having a hard time making bets right now, especially with the Unity failure. Another thing that hasn't been brought up yet is that it's kind of like an oncology, or it's kind of like the field of oncology. You are trying to kill certain cells while sparing other cells in a human. And in cancer, you can tolerate a fair amount of toxicity, right? Because these people may die in a short amount of time. You cannot tolerate that level of toxicity in a metabolic disease, for example. You can't have hair loss or mouth ulcers when someone's taking something for a metabolic disease and they're in their 30s. So I think for pharma to jump into this space through investments or deals, I think it's actually going to take a bit of time, and it'll be indication or therapeutic area driven once there's good data around a specific program and approach.
Can I add?
Yeah, please. I was noticing you and looking to you to respond. Go ahead, Marco.
No, I think that's an absolutely great point. And I think so the hypothesis, promise, and aspiration of senolytic as an indication, such as aging as an indication, which is not an FDA-approved indication yet, even if it's been considered like there is syndromes around that. There is a trial that people are trying to test a potential clinical path to look at the trajectory of aging, like using metformin, and that will be more a precursor of future clinical trials. But I completely agree. That's not the smartest things to do. Probably you first need to identify a specific clinical indication where senescent biology plays a driving critical role, show efficacy and success, and then take it from there. So for example, and we thought a lot about that, right? Senescence is a master regulator, is behind many potential, it's like integrating all this damage and driving all those diseases, has lots of opportunities. And for example, we publish a proof of principle using this prodrug. It's very rudimentary the idea based on what we're actually currently doing in terms of our lead compounds. But that showed promising, interesting results in geriatric mice. Those mice that not only live better and longer, but improve cognitive functions, improve regenerative capacity in the muscle. But how can you translate this in clinically? That's not probably the best things to try to go after dementia. It would be extremely complicated, expensive, and high risk of failure. So we, for example, decided to focus into certain indications such as idiopathic pulmonary fibrosis, where there is, and we partner with some of the best people out there. We have a partnership with the Cedars-Sinai Medical Center, and Professor Hogobam has been working in interstitial lung disease and senescence for 20 years or plus. It's clear how the role of senescence is a driver upstream to really this degenerative progressive events. It's a life-threatening condition. People have 2-3 years life expectancy, similarly to cancer. But we believe that if we could develop therapeutics that are safe and are disease modifying, so they can really be curative to a certain extent in those patients, will be a success for the entire field that can really enable have partners such as pharmas come to conviction that is actually a therapeutic opportunity. And so I completely agree, needs to be very strategic and carefully thought to make sure that we don't have clinical failures that can just cool down the enthusiasm, where in reality we're just at the beginning. But probably like in the 70s, where there was a twist in the oncology space, thinking of, oh, there's nothing you can do about it, to we can actually treat cancer. We can be very targeted. Now we're starting to have this revolution in the context of longevity, age-related condition, at least.
I completely agree with Marco.
Can I follow up quickly? I just had two things to add. So just, in regard to comparing cancer therapies to longevity therapies, I think with senolytics, the one advantage is you only have to give them once every 2 weeks, once a month, once every 3 or 4 months. So it's not chronic treatment. Cancer, you have to kill all the cancer cells or the patient will die. But for senolytics, it appears that even a 30–40% reduction has a positive effect. But having said that, our problem is we don't know what senescent cells we should target. And so actually NIH has put out a common fund from the director's office, $180 million to set up 6 centers to actually map the accumulation of senescent cells with healthy aging in humans, to identify what type of senescent cells are accumulating or maybe transiently accumulating in different tissues with age. So we'll then identify really what are senescent cells in different tissues, what biomarkers do they express, and then potentially develop ways to kill the ones which we think are actually driving disease. So we're at the very infancy of this field, because right now we're saying senescence, but a senescent podocyte is different than the senescent renal epithelial cell, different than senescent astrocyte, or a senescent lung fibroblast. They're probably all different and probably respond to a senolytic differently too. So, right now we're using a sledgehammer because we don't know what senescent cell to target for idiopathic pulmonary fibrosis or for Alzheimer's. But NIH is putting money into this field, and I think it's going to move the field forward very quickly.
Agreed. I think once the first small biotech shows efficacy in humans, I think that'll be the tipping point where pharma will start to get really interested. And again, it's so much like oncology. I mean, we used to use a sledgehammer of mustard gases and those kind of very toxic things. And now we're super sophisticated with antibodies that target very specific cells. And similarly, cancer is not just a single disease. There are multiple causes to different cancers. So lots of analogies can be drawn between the two. But yeah, I think a biotech will need to have established human efficacy and safety before pharma will make a big plunge. And again, I don't think pharma will plunge into senescence, though it'll probably be program by program that fits into their therapeutic areas. I could be proven wrong. Calico did start trying to tackle aging. But I'm not aware, but I'm not aware of any big pharmas who are focusing on senescence as a category.
And we've spoken to many big pharma, and I agree with you completely. A very accurate assessment of our discussions. They really focus on specific diseases and disease indications and not aging at this point.
Yeah, and it's unfortunate that Unity had that setback last year. But again, I feel like that was, those are sort of senescence 1.0, and I've seen some really sophisticated approaches recently.
And maybe to that point, I also want to kind of spend a word of support for Unity, because in reality, besides being amazing precursors and really supported the field when that started, I was super excited. It was predicted, and as we said initially, repurposing drugs for oncology space had limitations. Now, I think that the failure in clinical trials is not saying that is a failure for a senolytic approach or the role of senescence in a pathology. I think that's pretty clear, both from a preclinical and a clinical standpoint in many indications. I think the design of that specific clinical trial was very challenging, and if it was working, it has been fantastic. But a low single ascending dose with a 12-week time points for osteoarthritis, was a hard call. And unfortunately, in that case, you need luck, right? But maybe the treatment was working, simply has to be repeated, test different doses, different regime, but that's not how it works in clinical trials. So often you have only one shot. But based on all the preclinical data that I have, even the phase 1 was actually very promising. So I suspect that in reality, it was just an unfortunate conditions and events in the phase 2 that didn't give significance, statistical significance in their endpoints. but I still think that it was a promising strategy.
I agree. That's a very sophisticated interpretation of the outcome, but not everyone has that level of understanding, unfortunately.
True.
I tend to agree with Marco on that largely, because this has not been a setback for the field. It's not even been a setback for Unity, because Unity is bounced back. They've announced their phase 1 clinical trial results for another program, and their share prices are up already. So now I want the next question is for all the three participants and all the three panelists. What are the bottlenecks that exist today in the field of senolytics? What improvements have to happen to make it something that becomes a part of drug discovery?
So I'll start. I mean, there's probably a lot of bottlenecks, but I'll start with as the academician, which probably has different bottlenecks than Marco or Angela. But I think the biggest issue is really our lack of knowledge about what defines a senescent cell in vivo. And so trying to understand what we need to be targeting. And I guess the other thing I should have mentioned earlier is there are primary senescent cells, which are probably the drivers of disease. And then there are secondary senescent cells that are kind of the consequence of the drivers, but then they contribute. And we think that the primary cells that have most of the DNA damage or whatever event that drove senescence leading to the inflammation, the secondary senescence is going to be different. And really trying to understand what are the key target cells that we need to develop senolytics for. I mean, we're giving a senolytic systemically for 5 different indications. It's the same drug, but we don't know what the key target cells are. So, it's going to be, and you can't develop a drug unless you really know what the key target or senescent target cell is. And so I think there's just a lot of basic biology that still needs to be done, along with translational science. I think the clinical trials are great because they're proof of principle, but they're not optimized drugs optimized for different types of senescent cells. So, I still view this as there's a lot of basic biology about senescence, as well as translational work that needs to be done to really make this a clinical reality.
Paul, I just want to fold in the question from the audience about the biomarkers. So when you talk about differentiating different types of senescent cells, maybe primary, secondary, are you also bringing in the issues with lack of biomarkers, established biomarkers for studying these in the field?
Right. So, the field, the way it stands now, the biomarkers are identified by taking blood and seeing things that go up with age, and then trying to link them back to the senescent cell. And I guess the one thing that's coming out of the clinical trials is you can now look at these markers in serum plus or minus a senolytic. If those drugs are truly senolytic, it could still be in question, but plus or minus. So there have been proteins and lipids identified. Judy Campisi's lab, Nathan Leverzer's group, and others. There's evidence that there may be a senescent epigenetic clock that Morgan Levine is working on. So there are a number of different markers, and there seems to be lipids, there's DNA in serum that's being measured, mitochondrial DNA, a lot of different markers. But we don't know from what type of senescent cells they're coming from. You find them in serum, you can find them in urine, you might, in the context of senescence in the brain, actually find it in saliva. But we don't know what the senescent cell is that's giving rise to those. And that's where, hopefully, as we get more and more into the details of what senescent cells are arising with age and disease and characterizing them, we'll identify very specific biomarkers that can identify an increase in senescent neurons or senescent podocytes or senescent FAPs in muscle, whatever.
Yeah, Paul, I absolutely completely agree. Thank you for explaining this very well. And that's one of the reasons why, for example, the way we decided to approach this, and I realized that you need a number of resources that sometimes and speed. Academia sometimes is different. So what is less known in our approach that behind this pharmacology, this chemistry to make those prodrugs and others, we have a computational platform that we call Alembic, but all the names in a company from alchemy, by the way, it's really based on analyzing in a complex way. Our CTO is one of the former lead software engineers from Facebook who comes from a very different field. They just brought a very sophisticated way to analyze complex omics data from multiple angles, different types of omics data, from a single cell RNA-seq, proteomics, metabolomics, clinical data, across multiple dimensions like ages, which is important, disease, patient types, species, with the idea of triangulating and seeing what is emerging with age and disease. So how do we identify the pathological cells? In the case of senescent cells, what are the right senescent cells in the right time? How do they diversify? What do you want to target? So we're really spending lots of time and effort in answering to those questions, because you said, if you do the wrong hypothesis, you will figure it out later when you try in a clinical setting, and that's too late. I think the other important aspect is developing the right preclinical models, because to ask the right questions, you also need to write models to answer to that. And relying on many established models, even gold standards that are used for translate therapeutics, you need to be very careful. Sometimes we need to redevelop or think differently to really include aging into the equation and really include something that is predictive for a human pathology. Many results that or assumptions we do are based, for example, mice or rodents. They don't really necessarily tell us the story in humans. So I think it's very important also to build the right models to inform a clinical path.
Yeah, I agree with everything Paul and Marco said, and maybe just a couple other points. One, I think to Marco's comment earlier is also figuring out the right clinical trial design is probably key too. Again, it makes me think about the early days of cancer immunology, specifically when they were naively looking at the size of the tumor and whether it was shrinking over time. And they were finding the tumors were actually staying the same size or even growing bigger, but the patients were doing better and they couldn't figure it out. It's because T cells were moving into the tumors. And so they were staying the same size or even getting bigger. And so it could be that we need to rethink how the clinical trials are done as well for this field. And then the last thing I would say, which is more of a positive note, is that I think the one good thing that we have going is that there are so many different drug modalities available to us right now, more than ever. There's gene therapy, there is CRISPR, DNA editing, RNA modification, small molecules, antibodies. So on the one hand, we do have this big toolbox that we could use once we can identify the right targets. But getting to those right targets is going to be challenging.
Absolutely. And if I can add again, sorry, to what you just said, Angele, there is not one bullet kills them all. I think there are different modalities, there are different indications, and probably, the success of one of us will lead the success of everybody else. So it's very synergistic. And in the future, I can see synergistically many therapies targeting senescence can actually work together in combo therapies as we better learn and understand. So we need to think, and like cancer, it's a very sophisticated heterogeneous space.
Mm-hmm. Yeah, maybe we can fix your diabetes and your dementia all at once.
That'll be fantastic. Brandon, you have a question.
I do. And actually, before I do, we have looks like 9 minutes left, so maybe a couple more questions. I wanted to thank you guys. You're really a dream panel, the way that you're interacting so much among yourselves. So I really appreciate you. Okay, so this is kind of a lightning round that I'd love to get relatively quick answers from each of the three of you on. It's a two-part question. So there are a number of senolytics supplements available today. Just last night, I was kind of checking around online and found that there were a number of things that I could have ordered and had shipped to my home. So I'm curious, are any of you taking anything that's available right now in this space? That's the first question. So that can be a relatively quick answer. The second question is, more broadly, what are your top personal longevity recommendations that our audience can follow right now?
Good questions.
I'll start with that one, because I think most of the natural products that are sold as senolytics are based on fisetin, which was based on a paper that we published several years ago You can buy Seno Light and Seno this and whatever, and it's all Fisetin, and there's other ones coming. So there's going to be a variety of things you can get. They're all based on a flavonoid or combination of flavonoids. I don't think they're the most effective, but they do appear to work. They probably have a lot of other effects because flavonoids affect a lot of different pathways. So there's probably a lot of mechanisms through which they're working. When we showed now almost a year ago that giving old mice Fisetin reduced mortality after exposure to a coronavirus, I started taking Fisetin. So I started taking it in the kind of a dosing regimen similar to the clinical trial that's ongoing. Now, we don't know if it's working because it's a blinded trial, but I started taking it. Anecdotally, my knee with a little bit of OA started to feel better. Don't know if it's related to Fisetin. I didn't get COVID, which I don't know is related to Fisetin, but I started to take Fisetin every 2 weeks, kind of a high dose that we had shown works in mice. So.
You also look fantastic.
Well, thank you. I don't know if that's what it's supposed to be doing, but it's at least it's protected us, we think, from COVID, and maybe it's helping my right knee. So we'll see. But yes, and I know other people are taking Fisetin too that have worked on it. A lot of people take metformin. It's another one that a lot of people are out there taking, and then the NR, NMN.
Paul?
Yeah, but it's relatively easy to get. It's
okay.
Yeah, there's a lot of people that will prescribe that. It's because the price is, you get it basically for free at the pharmacy, but you need a prescription. But a lot of people are taking it for aging. And then NMN and NR, so the NAD supplementation. Obviously, Elysium has an NR based product, and then there are companies selling NMN to boost NAD. And I know a lot of people are taking those too. I don't like to take pills. So the only thing I'm taking at this point is Fisetin.
Paul, yes, you look fantastic. But besides, on top of that, I agree. I also want to spend a word of caution on so I think it's especially in US, supplements are not regulated. Like in Europe, for example, there are lots of side effects. So I would say people should be careful. And I do agree with actually I call it Fisetin. Now I see you saying Fisetin, but
I don't know the right. You're probably right.
I don't know the right. I always call it Fisetin.
I'm so glad you guys aren't sure how to say it, because that's why I said it.
Well, no, Paul is he is the one that published about it, so he decides the name. I'm going to change it like
it's tomato. It's I don't know what's.
But a word of caution, because I do think it's beneficial effect. I do think that also is aging related. So, for example, for younger people should probably be careful about using it. There are we generated data that a senolytic treatment taken regularly could actually have detrimental effects and completely opposite effects after a certain age. So when is the right time to take it? That's where we need biomarkers. There is a biological age. Some people might need it in the 30s, other in the 70s, and wow, how frequently, how long? I think those are questions we need to do. So that's why going first in a specific disease indication, patient population, and address this will inform how to expand this in a more regular preventative way. So I'm taking it too, just for the records, but in a very regulated way, in a very, like if I'm like sick or following a stress where maybe combined with some antioxidants to prevent certain damage. I don't think that, for example, for me, it would be necessarily beneficial to be taken on a regular basis. But we are more sophisticated. So I was going to say people maybe really consult with an expert to understand if and how you should take supplements. There are lots of things that might not be good for everyone or always.
So a quick question for you, Marco, there is, what is the concentration that or the dosage of Fisetin you're taking? And that's the same question for Paul as well.
I think I should interject. I said I take it, but I'm not, as Marco said, I'm not recommending people take it because we don't know the dose and we don't know long-term safety. So we're doing safety studies. So, it's a choice that I made to take it, but I'm not going to recommend dosing because I'm not sure what the right dose is. So, it's in clinical trials, it's in the nursing home study, it's in the COVID trial, it's in the Alzheimer's trial. So we will know a lot more about safety, hopefully in the next 6 months to a year. So I would not recommend people rush out and start taking massive doses of fisetin, thinking that if you take it every day, it's gonna be better than if you take it every week. So I'd rather not play doctor on this and recommend dosing.
Yeah, no, likewise. If anything, you ask what are the right, alternatives or healthy way. Exercise. There are plenty of literature showing that you can even reduce the senescent burden. In a healthy aged condition, a good healthy diet, healthy lifestyle, sleeping well enough. Those things are really showing that are the best way. Calorie restriction, there are open questions how to really translate from animal studies into humans. Us humans are so different that not everyone is the same. So I think, but really exercise is probably the best anti-aging practice that we currently have. And it requires more effort than taking a pill for sure.
Can I add a couple more comments too? So I'm also very leery of supplements because I know it's unregulated. So I take something that I think is probably fairly safe, and it's just curcumin, which is in food. So I take curcumin, and then on top of exercise, I would also say wear your sunscreen. Super important.
Curcumin has weak senolytic activity. It regulates NF-kappa B. Curcumin regulates NF-kappa B, suppresses SAS, so it probably has effect on senescent cells. Don't know if that's how it's working for you, but
I have a strong history of heart disease in my family, so I figure just reducing the inflammation is probably a good thing.
Awesome. Well, thank you so much for that lightning round. I think it went just as it did the last time I asked that question. It began with excitement, then it moved to caution, and then it closed with a recommendation to exercise and eat well, which is very important for all of us to remember. We love to end on time, so we will do that. We're coming right up on the hour. So let me, to close, just say how thankful we are to each of you, Angele, Paul, and Marco, for the great work that you're all doing to increase health span for all of us. We didn't all start at 5 years old on this mission like Marco did, but you are all today doing really, really amazing and important work. So thank you for taking some of your time to share with us and our audience. We really appreciate you.
My pleasure. Thank you.
Thank you so much.
Thank you, Jyothi and Brandon. Appreciate it.
Okay.
Have a wonderful day, everyone.
All right.
Bye-bye.
Bye-bye.
Take care.